Hello friends, welcome to BMH Learning. This video is regarding ligation using T4-DNA ligase. T4-DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in a duplex DNA or RNA. The enzyme repairs single-strand nicks in duplex DNA, RNA, or DNA-RNA hybrids. It also joins DNA fragments with either cohesive or blunt termini, but has no activity on single-stranded nucleic acids. T4-DNA ligase requires ATP as a cofactor. Protocol for running the experiment. Set up the following reaction in a microcentrifuge tube on ice. T4-DNA ligase should be added last. The T4-DNA ligase buffer should be thawed and resuspended at room temperature. Gently mix the reaction by pipetting up and down and microfuge briefly. For cohesive or sticky ends, incubate at 16 degrees overnight or at room temperature for 10 minutes. For blunt ends or single base overhangs, incubate at 16 degrees centigrade overnight or room temperature for 2 hours. Heat inactivate at 65 degrees centigrade for 10 minutes. Chill on ice and transform 1 to 5 microliter of the reaction into 50 microliter competent cells. Keep in mind these things while doing the experiment. Binding of T4-DNA ligase to DNA may result in a band shift in aqueous gels. To avoid this, incubate samples with 6x DNA loading dye and SDS solution at 70 degrees centigrade for 5 minutes or 65 degrees centigrade for 10 minutes and chill on ice prior to electrophoresis. The volume of the ligation reaction mixture should not exceed 10% of the competent cell volume in the transformation process. Prior to electrotransformation, remove T4-DNA ligase from the ligation mixture using spin columns or chloroform extraction. This was all. Thanks for watching.